Welcome everybody to Miltenyi's World of T-Cells, solutions to enhance your T-cell experiments. In the next 50 minutes, I will give you a short introduction to our company, the role of T-cells in general, and the concept of T-cell workflows, as well as pointing out our T-cell highlight applications. So let's begin with a brief company presentation. Miltenyi Biotec was founded in 1989 by Stefan Miltenyi himself and is the largest owner-managed company in Germany. We have two main sites in Germany, one in Bergisch Gladbach, where the headquarters are located, and one in Teterow, where the GMP manufacturing facility is located. We have subsidiaries in 28 countries and distribute to additional 73 countries. Currently, we employ over 3,000 employees worldwide. Our mission is to serve the needs in life science research and to translate new scientific findings into clinical applications. Let's have a brief look at T-cells and why they are so important. T-cells play as effectors of the immune system an integral role to the adaptive immunity. They can be subdivided broadly into several phenotypically and functionally distinct subsets, such as T helper cells, cytotoxic T killer cells, T regulatory cells, as well as the rather heterogenic group of unconventional T-cells comprising, for example, natural killer T-cells and gamma delta T-cells. T-cells are the only immune cells that are able to form a long-lasting immunological memory that helps to respond faster and more efficient to already experienced threats. Key surface markers are of course the lineage markers CD3 as well as CD4 and CD8, but also activation markers such as CD25, CD69, CD137 and CD154. T-cells communicate and exert their functions mainly via cytokines such as IL-2, IL-4, TNF-alpha and interferon gamma. T-cells are most relevant in both maintenance of health as well as fighting diseases. Last but not least, these properties make T-cells ideal tools for immunotherapy, where, for example, T-cells are genetically modified into CAR T-cells to attack specifically cancer cells. Although our best-known products are indeed the microbeads and isolation kits, we are not just a cell separation company. We actually cover the whole experimental T-cell workflow from cell separation to cell analysis, including also sample preparation as well as cell culture. We provide thereby solutions for each step of your experiment. So our T-cell portfolio is indeed large. One way to keep a good overview is to divide our T-cell products into our different main workflow technologies. Sample preparation, for everything that has to do with preparing your sample, for example tissue or blood, before you isolate your T-cells and you start your experiment. The gentleMACS tissue dissociator and the tissue dissociation kits are here the hallmark products. Cell separation, our classical microbeads and isolation kits in all their variants to isolate efficiently T-cells and subsets. Cell culture, everything needed to culture, activate and expand your T-cells, from media to cytokines to antigens. T-cell antibodies, flow kits, but also instruments for flow analysis, sorting and even imaging. Well, although it's now easier to get an overlook, we thought that maybe the best way to arrange our large T-cell portfolio are distinct workflows, specifically designed for your experimental needs. Thereby, we cover as focus workflows, amongst others, regulatory T-cells, CAR T-cell research, gamma delta T-cells, tumor-infiltrating leukocytes, polarization of T-cells, antigen and virus-specific T-cells, and of course, activation and expansion of T-cells. With these comprehensive workflows, you can easily choose the products relevant for your specific field of interest. Getting your whole workflow from Miltenyi Biotec means ease of mind. Standardization, reproducibility, and organization of an experimental workflow become much more feasible. Miltenyi Biotec as supplier for the whole T-cell workflow means that all reagents fulfill the same high-quality criteria and the order process can be streamlined, thereby harmonizing the steps of an experiment as well as automation and upscaling are easily performed. Getting all products from Miltenyi Biotec guarantees smooth procedures and less errors. Since, for example, our cell separation is tested for compatibility with our flow cytometry and our media for compatibility with our cytokines and so forth. Moreover, having Miltenyi Biotec as full supplier for the whole workflow means also getting full support for every step of your experiment, thanks to our great scientific support team. Moreover, a hallmark feature of Miltenyi Biotec's workflow solutions is the seamless translation from a research setting into a clinical setting and back, thanks to our MACS GMP portfolio, as well as manufacturing platforms such as the gentleMACS Prodigy. Well, let's start our journey through the T-cell workflow, taking the first step, sample preparation. Every successful experiment starts with a proper sample preparation, so start smart. So, your samples need to be prepared accordingly before starting your T-cell experiment. This is especially true when working with human and mouse solid tissue. Keep in mind, the quality of sample preparation influences all downstream applications. For example, a homogeneous and viable single-cell suspension is the basis for successful and efficient cell separation. Miltenyi Biotec sample preparation solutions, namely the gentleMACS tissue dissociator and the respective tissue dissociation kits, combine efficient automated mechanical tissue disruption with gentle and tissue-specific enzymatic digestion. With this, you obtain an optimal single-cell suspension with T cells of highest viability and preserved epitopes for the subsequent cell separation. Streamlined sample preparation with the gentleMACS ensures optimal reproducibility in downstream compatibility. The gentleMACS hereby is programmable, has optional automated heating, and can process up to 8 samples in parallel, eliminating thereby user variability. Here we have a good example of the efficiency of the gentleMACS dissociation. Preparing T-cell from mouse colons has been challenging as the commonly used protocol is labor-intensive and often results in low cell yield. Using the gentleMACS in combination with the laminar-propria dissociation kits enables to standardize the workflow and obtain consistently higher numbers of T-cells than with homebrew or manual methods. In the top left dot plots of CD4 and CD8 T cells, you see how efficiently the tissue dissociation of mouse lamina propria is performed with the gentleMACS and the respective enzyme kits. Well-distinct cell populations are visible in staining with CD3, CD4, CD8A, and CD49B antibodies, followed by analysis on the MACS quant analyzer, show a consistently high numbers of viable T cells. However, even more rare and delicate cells like Tregs can be prepared with maximum efficiency using the gentleMACS and the laminar propria kit, as you can see on the lower left. Two methods were compared. First, the gentleMACS protocol using the laminar propria kit, and then the gentleMACS respective program. And second, the manual method, which means cut the tissue into smaller pieces, add fetal bovine serum, add different enzymes, and incubate for an extended period of time. Subsequently, analysis of CD4 positive, FOXP3 positive Treg cells in the cell suspension was performed on the MACS quant analyzer. Analysis not only revealed that the efficient gentleMACS dissociation resulted in distinct and viable cell populations as seen in panel A, but also that consistently more Tregs are obtained comparing to the manual method as seen in panel B. The last example on the right shows preparation of tumor-infiltrating leukocytes from B16F10 melanoma mouse tumors, again comparing dissociation with the gentleMACS protocol and mechanical manual dissociation without enzymes. The cell suspensions were subsequently stayed with reaffinity recombinant antibodies for TIL markers such as TIM3, PD1, and LEX3. It can clearly be seen that the yield of viable target cells is up to 5 times higher using the gentleMACS system than with the competitor method. In summary, the gentleMACS and kits ensure high cell yield and viability even from hard-to-process tissue. As depicted in the small info box on the lower right, apart from the laminar propria mouse kit, we of course also offer other dedicated tissue dissociation kits to get highly viable T cells, from example from spleen, lung, liver, and tumors tissue. Now, once your sample is properly prepared, it is time for the next step, the separation of your T cells. Of course, here our renowned MACS technology is the way to go. So select the best for all your cell separation needs. Cell separation poses many challenges to the researcher. High purity and recovery are of course important, but also reproducibility, cell viability, and compatibility to downstream applications have to be ensured. Furthermore, there is a variety of different T-cell subtypes that might be of interest and have to be separated individually. Some of them display very low frequencies in the variety of different starting materials, hard to obtain. And of course, cell separation should be fast and easy to use. Consequently, there are many cell separation challenges and pain points to face. Gladly, Miltenyi Biotec offers the right solutions for all of them. Generally, MACS cell separation technology offers two different separation strategies. First, positive selection with our microbeads and microbead kits. Target cells are hereby labeled and directly selected their specific and defining marker. As target cells are still magnetically labeled after separation, they are usually not suited for an additional round of sub-separation, except when you use REAlease MicroBeads, where the label can be removed via an additional step. This positive selection approach via microbeads is very specific and therefore ideally suited for rare cells or undefined single-cell suspensions, such as tumors, as well as other difficult to or compromised samples, for example when working with patient material. Also, isolation direct from whole blood and blood products is very well achieved with MACS positive selection. Second, we have the untouched isolation with our isolation kits, working with the depletion of non-target cells. This is necessary when the target cell fraction is not defined by a certain marker, but rather the lack thereof. Hereby, all cells expressing defined exclusion markers are depleted and the target cells remain unlabeled and thus are usable for further rounds of sub-separation. However, in order to work efficiently, the single-cell suspension has to be rather well defined, as it is the case, for example, with human PBMCs or mouse single-cell spleen suspensions. Taken together, different isolation strategies allow to get the most out of your sample. With Miltenyi Biotec being an expert in all cell separation strategies, playing with and combining the different approaches offers a great flexibility according to your needs and is the secret to success. Consequently, MACS cell separation technology comes with 4 different approaches to perfectly reflect the different starting materials. First, we have microbeads and microbead kits that are ideal for positive T-cell selection from PBMCs and various tissue single-cell suspensions. They also include a new REAlease technology for label-free cell separation. Separation of basic T cells and almost any T cell subtype from different species like human, mouse, rat, and non-human primate make microbeads and microbead kits the most versatile cell separation approach. Second, we have isolation kits that are ideal for all untouched separation needs. The used starting material are PBMCs or defined single-cell suspensions. Also, the isolation kits cover basic T cells as well as almost any T cell subtypes from different species like human, mouse, rat, and non-human primate. However, if you do not want to go over the density gradient to get PBMCs as starting material, you can now also isolate T cells fast and directly from whole blood and blood products. So, as third option, we have our StraightFrom MicroBead Kits for the isolation of human CD3, CD4, and CD8 T cells directly from whole blood and blood products without the need for density gradient centrifugation. It is a reliable way to get your T cells fast and easy and, of course, automatable. As fourth separation variant, we offer MACSexpress isolation kits. Also here, human Tregs, pan T cells, as well as CD4 and CD8 subsets and even CD4 memory T cells can be isolated directly from whole blood and blood products without the need for density gradient centrifugation. This time, however, it is an untouched cell isolation approach. MACSexpress is not automatable though, but indeed very fast and uncomplicated. One of the many things that sets Miltenyi Biotec's T-cell portfolio apart is its broadness and diversity. We cover more than just the basic human and mouse T-cell subtypes. Indeed, over 70 T-cell separation products cover virtually every T-cell subset, addressing T-cells researchers' needs worldwide. For example, different naive and memory T-cell subtypes as well as T helper cell subtypes are covered. Furthermore, different T regulatory cells, gamma delta T cells, and NKT cell separation kits are available, as well as complete workflows for antigen-specific T cells, cytokine secreting T cells, and CAR T cell research, and many, many more. Apart from human and mouse samples, we also offer T cell separation kits for rat and non-human primate samples. As scientists, we understand how important it is to address the full complexity of T cell subtypes, even for the rare ones, and to offer according solutions. In the following slides, I will give you an overview of the most important T cell separation products, including our highlights. But why use Miltenyi Biotec for cell enrichment, and what does make MACS cell separation technology so special? In short, our select the best approach. Minimal labeling with no effects on cell biology for full downstream compatibility. In more detail, this is due to the columns as technological advantage. The columns amplify the magnetic field by 10,000-fold. This allows for minimal labeling and smallest bead sizes of only 50 nanometers. In consequence, less epitopes are covered on the cell surface and less stress is applied on the cells. So, as thousands of publications prove, the isolated T cells are fully suited for any downstream application, including flow analysis. Thereby, MACS microbeads were even approved by the FDA to be used in a specific clinical setting. It is important to note that the column allows for the most gentle sorting, since there is a large space between the steel spheres of the column matrix with about 20 times the size of a lymphocyte. The T cells will pass through smoothly without any negative effect. Have a look at the data on the right side with CD3 labeled T cells. Column-free technology results in bead aggregates in cell culture and massive overlabeling on the cell surface. With MACS column technology, you see no bead aggregates and minimal labeling for fully functional cells. And indeed, you see also in these Western blots that T cells are not activated after cell isolation with MACS technology. CD4 T cells were isolated with either positive selection or negative selection using MACS technology or column-free competitor methods. Their activation status was then assessed by lysing the different isolated T cell samples and performing a Western blot for MAP kinase phosphorylation, a reliable activation status marker. On the left side, you see a Western blot for p38 phosphorylation after positive isolation of CD4 T cells using either Miltenyi CD4 microbeads, human, or two column-free competitor systems. Untreated and LPS-treated SUPT1 cells served as negative and positive control, respectively. It can be clearly seen that cells isolated with MACS technology yield a much lower phospho-p38 expression than cells isolated with the competitor's column-free technology. On the right side, you see a Western blot for JNK1 and 2 phosphorylation after negative isolation of CD4 cells, T cells using either Miltenyi CD4 T cell isolation kit human or 3 column-free competitor systems. Also here, untreated and LPS-treated SUPT1 cells served as negative and positive control, respectively. Again, it can be clearly seen that cells isolated with MACS technology yield much lower phospho-JNK kinase 1 and 2 expression than cells isolated with the competitor's column-free technology. Both Western blots clearly show that cell separation with MACS technology is the most gentle one and does not activate T cells, regardless of positive or negative selection. The first highlight cell separation products are our T cell isolation kits for the untouched isolation of various human and mouse T cell subsets. Of course, the kits offer our well-known high purity in recovery. However, also a very important factor is of course the protocol speed. Especially with untouched isolation, faster protocol times are favored. Miltenyi Biotec offers therefore a total of 17 fast cell isolation kits that allow to isolate publication quality target cells in only 18 minutes. The 6 mouse fast isolation kits comprise the most important T-cell subsets as well as 2 B-cell kits. The 11 human fast isolation kits also cover the most important T-cell subsets as well as B cells, NK cells and monocytes. So instead of long and tedious processing, we offer fast and easy protocols. However, it does not stop with the isolation kits. Also, our best-selling mouse microbeads were improved and the protocols updated by reducing tedious washing steps and optimizing incubation times. In 3 easy steps, you get from spleen to target cells in less than 15 minutes. Simply straightforward. CD4 and CD8 mouse T cells, as well as CD90.1 mouse and rat T cells, and also CD90.2 mouse T cells, can now be isolated via positive selection in only 13.5 minutes using our updated MicroBeads mouse. Of course, with the ever appreciated quality and purity. It is an easy and fast protocol that can be fully automated on the AutoMACS Pro or the MultiMACS. Each MicroBead kit has a high capacity for 2 times 10 to the 9th total cell, which corresponds to up to 20 mouse spleens. Consequently, with the high sample capacity and automation options, the updated MicroBeads mouse are not only economic but also very efficient and therefore ideally suited for the processing of high sample numbers. Have a look at the product page by following the links above. Here you will find scientific posters highlighting the advantages of MACS microbeads, for example in mouse adoptive T-cell transfer experiments. And again, it does not matter if you use positive or negative selection. Cell separation with MACS technology is fully downstream compatible and has no effects on cell biology in either case. Here T cells were either positively selected with CD4 or CD8 microbeads mouse or isolated untouched with our own respective T cell isolation kits. Directly after isolation, day 0, and after 1 day of cultivation with and without stimulation using the T cell activation expansion kit, marked with plus and minus respectively, activation markers CD25 and CD69 were analyzed by flow cytometry to assess potential activation. As you can see, without stimulation, cells are neither activated when separated positively with the microbeads, nor when isolated untouched with the isolation kits. This means that both cell isolation methods are equally gentle. When stimulated on purpose with the T-cell activation expansion kit, cells are highly activated and upregulate accordingly CD25 and CD69 regardless of the isolation strategy, meaning that both isolation methods are equally suited for functional downstream applications. Consequently, both approaches do not affect cell biology and ensure full T-cell functionality while ensuring fast and gentle isolation. As a useful tip, we observed that red blood cell lysis indeed activates T cells. However, red blood cell lysis is not needed for T cell isolation with MACS microbeads from mouse spleen, so better avoid it. Our next highlight product are the StraightFrom MicroBeads. StraightFrom MicroBeads allow the isolation of target cells directly from blood and blood products without density gradient centrifugation. In regards to T cells, CD3, CD4 and CD8 positive cells are covered and can be directly isolated from whole blood bufficoid leukoreduction system chambers, abbreviated LRSC, and leukopecs. Of course, cell separation with StraightFrom is automatable. This means processing on either AutoMACS Pro or MultiMACS are possible and reduces processing time, for example, of a whole bufficoid to only 30 minutes. Indeed, protocols need to be fast and easy, especially for larger screening purposes. Automation and high throughput options may be desirable. Furthermore, starting materials, in this case blood products, come in different formats, so a restriction here is not desirable. Moreover, Sometimes samples and cell numbers are limited, so of course cell loss, especially when working with small or rare samples, must be minimized, but purity maximized. However, most importantly, cells must not be activated by isolation to ensure that, for example, really only the specifically stimulated T cells proliferate and are fully functional. StraightFrom is the ideal solution here. A fast and flexible positive isolation approach that allows to isolate different cell types from the same sample with minimal loss due to the skipping of density gradient centrifugation. To date, the most widely used method to isolate target cells from whole blood and blood product is, however, performing still density gradient centrifugation for the generation of PBM cells first, followed by cell separation of the target cell subtypes. Apart from the time-consuming protocol with over 2 hours processing time, the handling is rather complicated and does not allow for automation. Furthermore, a significant amount of target cells may be easily lost in a user-dependent fashion. In addition, harsh and long handling as well as some gradient media may also affect the cells, at least to some degree. So act carefully. The alternative to density gradient centrifugation is Miltenyi Biotec's StraightFrom technology. Hereby, cells can be directly isolated from blood products in a fast and easy way, without density gradient centrifugation, cell counting and washing. The procedure is fast but gentle and can be fully automated. So, from sample to target cells in only 30 minutes processing time. This is of course also backed up by flow cytometry data and assessment of activation marker expression. CD4 and CD8 T cells were isolated from a Buffy code either with StraightFrom CD4-CD8 microbeads on the left side or via density gradient centrifugation, followed by a competitor's untouched pan T cell isolation on the right side. Cells were analyzed for activation marker expression of CD25 and CD69 either directly after cell isolation, day 0, or after 1 or 2 days of cultivation, either without stimulus or stimulated with the T cell activation and expansion kit, labeled with minus or plus respectively. Cells isolated with StraightFrom show almost no activation marker expression even after 2 days of culture. However, when activated on purpose using the T cell activation expansion kit, the cells highly upregulated CD25 and CD69, showing their biological functionality. Cells isolated via density gradient centrifugation, followed by a competitor's untouched pan T cell isolation, instead show activation marker expression from the beginning on, and consequently may not be efficiently activated in downstream applications, such as polyclonal or antigen-specific activation. Our next T cell separation highlight are the REAlease MicroBead Kits. This revolutionary microbead technology allows to positively select cells, but then to remove the microbeads and the antibody labeling to get completely label-free cells. REAlease MicroBead Kits are available for many human and mouse T cells and non-T cell subtypes, including markers such as CD3, CD4, CD8, CD19, CD25, CD56, CD62L, CD90.1 and 2, PD1, and TCR gamma delta. This offers researchers an entirely new level of flexibility and new separation strategies, combining the best of two worlds. Highly specific positive selection, but if needed, label-free, for example for further subset separations. The procedure itself is quite easy. After cell separation, there is a bead removal step with the bead release reagent. Cells are now ready for a second round of magnetic labeling. An additional optional step can be applied using the REAlease reagent to also remove the bound antibodies, thus to obtain completely label-free cells and make all epitopes accessible again. The advantages of REAlease do not end here. REAlease is so gentle and minimally invasive due to the special structure of the REAlease complex that even cell isolation via CD3 does not activate the T cells, regardless of the label removal. as you can see in this graph of isolated human CD3 positive T cells on the left side. Activation marker CD69 is not upregulated amongst pre-gated CD4 cells, neither after CD3 labeling, nor after isolation, nor after label release. However, when activating the cells on purpose, CD69 is nicely upregulated. Please note that pre-gating on CD8 positive cells showed the same results. The example on the right shows a multi-step subset isolation using REAlease. First, CD4 T cells are pre-selected. Then the label is removed and cells are ready for a second round of deeper subset separation. In this case via CD25 for Treg isolation. Further examples include gamma delta T cells as well as CD3 negative NK cells. Finally, I want to point out a cell separation product that is absolutely unique, the Cytokine Secretion Assays. They allow to detect and enrich viable cytokine secreting cells from mouse and human samples. Cytokines are immobilized on the surface of the secreting cell by the so-called catch reagent. Basically, the cell labels itself with its own cytokines. In a second step, the immobilized cytokines are then labeled with fluorophore conjugated antibodies called detection reagent and can then be detected viably without intracellular staining on any flow cytometer. In an optional third step, the fluorophore-labeled cytokine secreting cells can be specifically enriched and used in downstream assays by using anti-fluorophore magnetic beads. Consequently, our cytokine secretion assays come in two versions. For flow detection only, we have the detection kits. For the separation of viable cytokine secreting cells, we have the detection and enrichment kits, including the additional enrichment step via a MACS column. MACS technology offers, depending on the researcher's needs, different manual and automated cell separation options for upscaling. Manual separation with the Quadra and OctoMACS for up to 4 or 8 samples in parallel, respectively. Automated upscaling options include the AutoMACS Pro for full walk-away convenience for up to 5 samples. The MultiMACS Cell 24 Separator Plus for the processing of up to 24 samples in parallel. And finally, the MultiMACS X for flexible, fully automated, high-throughput industrial applications with any source material. Now every researcher can reliably automate cell separation for more efficient workflows and higher consistency. Of course, the automation options are applicable to most of our T-cell separation solutions. Now, with successfully isolated T-cells, it may be time for the culture step of your experiment. So cultivate your vision. When culturing T-cells, apart from activation and expansion, also differentiation into different T-cell subtypes is an important topic. The latter is performed with cytokines and functional grade antibodies. Activation is performed with either polyclonal stimulation reagents that, for example, engage CD3 and CD28, or antigen specifically, using, for example, our Pep Tivator Peptide Pools. Expansion and culturing is best performed in special T-cell media, such as our serum-free therapeutic. In the following slides, we will have a more detailed look at Miltenyi Biotec's T-cell culture portfolio. Of course, culture media play for the cultivation of T-cells a pivotal role. Here we offer our serum-free therapeutic T-cell medium. Free of animal-derived components, it is available from research grade up to max GMP grade and ensures optimal T-cell growth, activation and expansion. Tedious pretesting of expensive and rare fetal bovine serum is not needed, and undesired side effects caused by the serum are eliminated. Originally, TCAEK was developed for the clinical grade serum-free manufacturing of human CAR T cells. However, TCAEK in research grade is of course also beneficial for many other T-cell applications, especially human ones. Moreover, for the polarization of mouse T-cells, TCAEK has been proven to yield superior results when compared to standard basal medium. The serum-free TCAEK T-cell medium allows for most consistent T-cell culture results, With the Max GMP version and of course with our expertise, translation into clinics has never been easier. Talking about T-cell activation and expansion. Our classical bead-based expansion reagents, the T-cell activation and expansion kits, offer a reliable yet flexible polyclonal T-cell activation. Of course, specific T-cell activation and expansion kits for mouse and human T-cells and Tregs are available. On top, we also offer a non-human primate version. The employed MicroBeads can thereby be flexibly loaded with biotinylated antibodies of choice to optimize activation stimuli. As seen in the graphs, both mouse and human expansion rates surpass very well 100-fold for both CD4 and CD8 T cells. T-cell activation and expansion kits are not only consistent in their performance, but also very economically priced, especially when considering the high cell capacity per kit. Our next polyclonal T-cell activation and expansion reagent is the new and absolutely innovative T-cell Transact Human. It is a polymeric nanomatrix that efficiently and reliably cells CD3, CD28 engagement, as you can see in the dot plots on the right, displaying strong upregulation for activation markers CD25 and CD69 in both CD4 and CD8 T cells after stimulation with Transact. As a nanomatrix, Transact is as convenient as it gets. Ready to use, easy volumetric dosage, and simple removing by washing. So no tedious debeading step is necessary, which makes Transact compatible to automated cell manufacturing systems and minimizes cell loss. Being available also in Max GMP grade, Transact is the ideal T-cell activation reagent for automated and streamlined translational applications such as CAR T-cell manufacturing. So T-cell Transact resolves quite some pain points right away. It's easy to handle, reliable and consistent in performance, as well as easy to translate into clinics. For the best activation and expansion, our specific T-cell medium TCAEK and our efficient activation solutions, namely the T-cell activation expansion kits and T-cell Transact, go of course hand in hand. As you can see on the left, TCAEK outperforms serum-free competitors and RPMI alike in a 14-day human T-cell culture after activation with the T-cell activation expansion kit, reaching over 80-fold expansion without the addition of serum. On the right side, you see how efficiently TCAEK and T-cell Transact combined work in a completely serum-free, fully translational cell culture setting. After 14 days of culture, human T cells activated with T-cell Transact in a serum-free TCAEK even outperform a competitor's classical bead-mediated T-cell activation in RPMI with 10% fetal bovine serum, being thereby more reproducible, convenient, and translational. Our third and last option for polyclonal T-cell stimulation is Cytostim, the non-toxic Staphylococcus enterotoxin B, abbreviated SEB, alternative. Cytostim cross-links the T cells TCR to the antigen-presenting cells MHC. This means, as with SEB, the T cells need the presence of antigen-presenting cells to be stimulated properly with Cytostim. This is best achieved in a PBMC setting and may serve, for example, as a functional control in antigen-specific T-cell experiments. Since the receptor cross-linking of Cytostim is antibody-mediated, no toxic superantigen like SEB is involved. However, the efficiency is very well comparable to SEB, as you can see very well in the dot plots below. So with Cytostim, no complicated to handle and dangerous reagent is needed, but we offer a simple to use and efficient alternative. Another way to activate T cells is the antigen-specific stimulation, for example using peptide pools. Please remember, this also works best in the presence of antigen-presenting cells, for example in a PBMC context or in a co-culture of T cells and antigen-presenting cells. Hereby, the most efficient antigen-specific stimulation of T cells is achieved reliably with our Pep Tivator Peptide Pools. These extensive panels of virus, tumor, fungi, microbiota or autoantigens consist of 15 mere peptides with 11 amino acid overlaps, covering the complete sequence of the respective antigen. The peptide composition ensures the optimal stimulation of both CD4 and CD8 T cells. Pep Tivators are available in different quality grades, as well as in a convenient 96 well format for high throughput applications. The different quality grades include research grade, premium grade, and max GMP grade. Most notably, we recently added SARS-CoV-2 Pep Tivators to our portfolio to support your research best in these difficult times. Please follow the respective links to get to the Pep Tivator product pages as well as the SARS-CoV-2 Pep Tivator video. Please note that we also offer TLR ligands as well as recombinant full proteins for cell stimulation. To stimulate and differentiate T cells, Miltenyi Biotec offers also MACS cytokines. They come in 3 different quality grades: MACS GMP grade, premium grade, and research grade. Research grade is the standard quality format and enables reliable and cost-effective studies with human and mouse and rat cells. Use premium grade cytokines for most consistent cell culture results. Premium grade cytokines offer the convenience of well-defined lot-specific biological activities in international units according to WHO standards. Thereby, they allow for exact unit dosing and eliminate laborious lot pre-testing. This saves money, but also time. MACS GMP cytokines come with extensive documentation, lowest endotoxin content, and highest purity, ideal for relevant preclinical studies and an easy translation into clinics. So indeed, standardization is the key for reliable cell culture results. Talking about convenience and consistency, the CytoBoxes mouse are kits that allow for the easiest polarization of naive CD4 positive mouse T cells into Th1, Th2, and Th17 subtypes without complicated ordering and calculation of single polarization cocktail components. The CytoBoxes contain in one convenient kit everything needed for the efficient mouse T helper cell polarization, including the cytokines and the functional grade antibodies. As you see clearly in the presented data, Th1, Th2, and Th7 cells are successfully polarized and express their respective signature cytokines and transcription factors such as interferon gamma, IL-4 and IL-17, as well as Tbet, GATA3 and ROR gamma T respectively. As you can see below, best results are of course generated with our special T-cell medium TCAEK. Our final chapter covers T-cell analysis. The method of choice is of course flow cytometry for assessing T-cell surface markers, secreted cytokines, and transcription factors. Miltenyi Biotec's extensive flow analysis portfolio not only includes antibodies, but also flow kits, cytometers, sorters, and even imaging system, satisfying hereby every researcher's need. So join the flow revolution. Our first highlight product in the flow chapter are the reaffinity recombinant antibodies. Being recombinantly engineered, we have full control over their genetic sequence and can express the reaffinity antibodies in a mammalian non-hybridoma cell system. This means that reaffinity antibodies exhibits highest purity and lot-to-lot consistency for even greater reproducibility, that they can undergo a most stringent in-house validation, and that there is no need for tedious and costly Fc receptor blocking, and require only one universal isotype control for maximum convenience and cost saving. Reaffinity antibodies are hereby readily available in hundreds of specificities and are best combined with our proprietary Viodye fluorescent antibody conjugates. Thereby, Viodyes are bright with superior mean fluorescence intensity for excellent signals, distinct with a high staining index for clear population discrimination, and hassle-free, since they are ideal for multicolor experiments due to low compensation. All this makes reaffinity antibodies the ideal choice for any flow analysis, since traditional hybridomas are not standardized by nature. Here, I'd like to show you very briefly just some exemplary flow antibodies that you can choose from to create your own panel, depending in which context you want to analyze your T cells, be it development, activation, exhaustion, or any other phenotyping. With our convenient antibody panel builder, this goes even easier. Just follow the link below and try out this nifty web-based software tool. However, we do not only offer antibodies, but also convenient, ready-to-use flow analysis kits, such as the Rapid Cytokine Inspector kits, as all-in-one no-worries solutions that combine surface marker staining with intracellular cytokine staining for the analysis of activated CD4 and CD8 T cells in the fastest way possible. MACSplex cytokine kits for determining concentrations of up to 12 different secreted cytokines in cell culture supernatants. And finally, the 8-color immunophenotyping kits for flow evaluation of various leukocyte subsets, for example, monocytes, neutrophils, eosinophils, and T, B, and NK cells. Importantly, all of these kits are compatible with the fast and convenient express modes on the MaxQuant analyzers. These software modules automate analysis and thereby not only speed it up, but also eliminate human error thanks to automated gating and analysis templates. Make sure to have a look at the new promo video for the flow kits, as well as the Express Mode and MaxPlex webinars by following the respective links on the right. One of our highlight flow analysis kits is depicted here, the SARS-CoV-2 T-cell analysis kit Human. This all-in-one kit allows the efficient and sensitive analysis of SARS-CoV-2 reactive T cells in human PBMC samples. After stimulation with our SARS-CoV-2 Pep Tivator Peptide Pools, cells are stained with an optimized flow panel depicted on the upper right side and analyzed, for example, on our MaxQuant flow analyzer. The kit includes a SARS-CoV-2 Pep Tivator of your choice, as well as the whole antibody panel and all needed buffers and controls. As you can see, we can beautifully detect, for example, interferon gamma expressing SARS-CoV-2 reactive CD4 and CD8 T cells after stimulation with a mix of our SARS-CoV-2 peptivators. Other markers, as depicted in the panel, of course, can also be assessed. Our flow portfolio is rounded off with our flow analysis and sorting instruments of the MaxQuant series. First, we have the MaxQuant Analyzer, powerful benchtop flow cytometers with up to 16 optical parameters. The clue here is that they allow for full automated acquisition using a robotic sample collector with the express modes, software modules, even data analysis is automated and free of human error. The small benchtop cytometers fit easily under the flow hood, adding further to operator safety, for example, when working with contaminated samples. Second, we have MaxQuant cell sorter. Opposed to classical droplet sorters, the MaxQuant Taito allows for the most gentle cell sorting and highest viability even for rare and fragile cells, thanks to its unique microchip sorting technology. Furthermore, the closed and sterile cartridge system makes the Taito the ideal machine for a GMP environment. In this context, I'd like to have a closer look at our MaxQuant Taito cell sorter, the gentleman under the cell sorters. With its unique capabilities, namely the most gentle sorting and the closed sterile cartridge system, the Taito is ideal for a GMP clinical environment, not only in the fields of CAR T cells and Tregs, but also for antigen and virus-specific T cells. So let me point out that the Taito is already part of several clinical cancer trials involving antigen, or better said in this case, tumor-specific T cells. Worthwhile mentioning is of course also the Taito's safety in virus research. The Taito works without internal fluidics and applies a sterile and closed cartridge system for maximum operator safety. Sorting purity is of course guaranteed, as you can see in the example below, where CAR-positive T cells were sorted after expansion and staining with our CAR detection reagent to over 99% purity. But the instrument highlights do not end here. A really fantastic novelty in our portfolio are our new imaging systems, MACSima and the UltraMicroscope Light Sheet Microscope family. These systems allow the visualization of T cells and others within a tissue context. Thereby, new CAR T targets may be identified, the distribution dynamics of TILs in tumor tissue assessed, and a deep phenotypic analysis of patient samples and the tumor microenvironment is possible. The MACSima Imaging platform for ultra-high content imaging hereby applies the renowned MIX technology. Spatial immune cell distribution, including their phenotypes, is assessed within tissue and cell conglomerates by sequential staining of hundreds of markers. In contrast, traditional staining techniques allow maybe the staining of 3 to 6 markers, giving only an incomplete picture. This analysis of hundreds of markers within a single sample includes, of course, automated sample processing and imaging of all types of fixed samples from tissue to single cells. On the other hand, the light sheet microscopes of the UltraMicroscope family enable you to visualize whole biological systems in 3D. Moving the sample through the light sheets allows the generation of sharp and bright 3D images of, for example, the tumor microenvironment, thereby really giving full insight into the tumor immune cell interactions. Please have a look at this impressive video taken with the UltraMicroscope 2. It shows a mouse xenograft of a human pancreatic carcinoma cell line with infiltrating CAR T cells in 3D. As mentioned before, the variety of our T-cell products is best organized in comprehensive workflows following your scientific interest. Consequently, please find here listed our highlight application workflows. Our highlights include polarization of T-cells with protocols and cytokine combinations for the most efficient polarization of your CD4 positive T helper cells. The regulatory T-cell workflow with all needed solutions for the isolation as well as the expansion and analysis of rare Tregs in a translational setting. The CAR T-cell research workflow covering all steps from T-cell isolation to activation to transduction and finally CAR T potency analysis for fully translational and functional generation of CAR T cells in a research setting. The TIL workflow to efficiently dissociate tumor tissue and isolate even the rarest tumor-infiltrating leukocytes. The antigen-specific T-cell workflow, optimized for the specific stimulation of antigen-reactive T cells, as well as the isolation and analysis of these extremely rare cells in different contexts. And finally, the COVID-19 virus-specific T-cell workflow that covers each step from stimulation to enrichment to analysis of SARS-CoV-2 reactive T cells, especially important in the current situation. Although that was quite an extensive overview of the T-cell portfolio, you have to remember only 3 easy things. We offer more than just T-cell separation. We cover the whole T-cell workflow, and we offer a solution to every pain point. So have all the success with our innovative and complete T-cell portfolio as displayed here in this full-featured overview. Before we end, please acknowledge the displayed disclaimer. Hereby, I close with this nice picture of our company campus in Bergisch Gladbach at night. Please visit our website www.miltenyibiotec.com to get more info on the mentioned workflows and products in the forms of application pages, protocols, and of course in-depth webinars. So thank you very much for your attention.